Hemoglobinopathies. Relating structure to function, types of protein, globular proteins, e.g. enzymes and hemoglobin. They are compact, soluble, hydrophilic residues outside and hydrophobic residues hidden. Fibrous proteins, e.g. keratin for hair and nails, and collagen. They're elongated, often have repeating amino acid sequences, and are insoluble due to high hydrophobic amino acid content. And then there are membrane proteins, e.g. ion channels. Abnormal hemoglobins. Over 400 mutant hemoglobins are known. 95% are due to single amino acid change. Hemoglobinopathies, some fatal, some advantageous, and some with no clinical effect. Why can altering one amino acid cause such a change in the structure and/or function of hemoglobin? The relationship between structure and function. Oxygen-binding proteins. Why do we need them? Cells require oxygen for oxidation of foodstuffs. Oxygen is only sparingly soluble in blood. Therefore, it needs transport and storage systems. Myoglobin is an oxygen store. Hemoglobin is for oxygen transport and carbon dioxide transport. Myoglobin stores oxygen in the tissues. It has the highest concentration in skeletal and cardiac muscle. It's a small protein with an MR of 17,800. It has 153 amino acids. It's very compact. It contains a heme prosthetic group. A typical globular protein, mostly alpha helical, with 8 alpha helices. It has hydrophobic residues on the inside, polar ones on the surface. The heme prosthetic group. Heme, Fe2+, and protoporphyrin, IX. Its tightly bound, non-polypeptide unit is essential for biological activity of the protein, non-covalently bound by a hydrophobic crevice in the protein. Fe2+ can coordinate with 6 ligands, 4 nitrogen from porphyrin ring, nitrogen from proximal His, F8, and an oxygen atom. Oxygen binding changes position of Fe. Distal His, E7, is also involved on oxygen binding. Advantages of associating heme unit with protein molecule. Fe atoms are kept in Fe2+ form. Binding of other small molecules, e.g. carbon monoxide, is inhibited. Carbon monoxide causes more than 50 deaths in the UK each year. Most result from faulty heating appliances. Carbon monoxide binds to heme with 250-fold greater affinity than oxygen, so prevents oxygen binding. Symptoms include nausea, dizziness, and confusion. Hemoglobin. 4 polypeptide chains held together by non-covalent interactions. Adult hemoglobin has 2 alpha and 2 beta chains. Each chain contains a heme group. Therefore, hemoglobin can bind to 4 O2s. 4 chains packed tightly together in a tetrahedral array with overall spherical shape, quaternary structure. Each chain of hemoglobin has a very similar 3D structure to myoglobin, but differ at 83% of amino acid residues. Very different primary sequences can specify similar 3D structures. One subunit is good, but 4 subunits are arguably better. This is because in hemoglobin, the oxygen binding is cooperative, whereas in myoglobin, oxygen binding is non-cooperative. In hemoglobin, oxygen affinity is affected by pH and CO2, whereas in myoglobin, pH and CO2 do not affect oxygen affinity. Also, with haemoglobin, oxygen affinity is regulated by BPG, whereas in myoglobin, oxygen affinity is not regulated by BPG. Myoglobin has a greater affinity for oxygen than haemoglobin. Binding of oxygen to haemoglobin is cooperative. The binding curve is sigmoidal. Binding of one oxygen makes binding of the next easier. Oxygen binding alters the tertiary and quaternary structure of haemoglobin. The effects of oxygen binding to one haemoglobin subunit. Proximal His F8 is pulled in, shifts helix F, EF, and GF corners. Altered shape transmitted to subunit surface. Some interchain salt bridges rupture. The effect of oxygen binding to one haemoglobin subunit. Proximal His F8 is pulled in. It shifts the helix F and EF and FG corners. The altered shape is transmitted to subunit surfaces. Some interchain salt bridges rupture. As the subunit changes shape, it therefore changes the relationship between the four subunits. The alpha-beta pairs slide and rotate relative to one another. These structural changes increase the affinity of the remaining subunits for oxygen. Change in the relative position of haemoglobin subunits affects oxygen binding. The T state, or tense state, has more salt bridges between subunits. It's got a low affinity for oxygen. The R state, or relaxed state, has fewer salt bridges with a higher affinity for oxygen. The Bohr effect shows changes in pH.